Congratulations on your presentation. I would like to invite to the stage Tamiris, Azamor, da Costa Barros, to present the poster Type 1 interferon innate errors causing severe adverse events following yellow fever vaccination, a family-based case study.
Bom dia.
Oh, good morning. My name is Tamiris. I work in the immunological laboratories in Miomonguinhos, and I'm going to talk about our study, which is type 1 interferon innate errors causing severe adverse events following yellow fever vaccination, a family-based case study. So the yellow fever is a gold standard for safety and also in terms of immunity. And it's very common in Brazil and other countries of Africa because we have the wild yellow fever virus and we had death cases in São Paulo. The immunology involves certain events that happen days after the vaccination, where you have the viral replication that is self-limited with an innate response, innate immune, and you have the development of a viral response and tumoral response very efficiently. And these events, they happen in this interval of days after the vaccination. So they're characterizing events that are previous to the vaccination, and also a high viral yield and hyperinflammation of the patients. The adverse events are classified such as neurotropics and viscerotropic diseases, and the viscerotropics happen in 2 to 5 days post-vaccination, and the mortality is 60%, and neurotropics has a higher range and also a lower mortality. So the objective of this study was to elucidate the mechanisms behind the pathogenesis of the adverse events after vaccination and to identify biomarkers that are useful to reduce the incidence of these rare events. For that, we use samples of a phase 4 national-based studies, and these studies counted on 5 cases, including the patients and their family members. So we have here in this table the 4 cases that we have, 1 neurotropic and 4 viscerotropic, and these 3 of them were also identified as innate errors of type 1 interferon, and they were published, especially developed by the group of Dr. Jean Casanova and Carlos Reif at the Rockefeller University. And these, just to mention a little bit, the interferon type 1, where it starts in the binding of the receptors of interferon 1 and 2. It's the main pathway for this process. And the patients that we described previously, we see an illustration where we found a truncation in the receptor and also loss of axon 2, and also patients that had high antibodies against interferon, and these led to this reaction. So in this project, we seek to analyze one of the cases that was not investigated yet, which was case 2, and we then went to the whole exome sequencing and validation of the findings and of PCRs of the cases and the family members. We also use a functional analysis, including the 5 events and also healthy controls. And we had the blood samples. We isolated the PBMC, and were able to put the samples with the virus, and we used immunophenotyping and other processes. And here we have the radiogram of the family. In this case, it's a family that have 11 siblings, and 3 presented adverse events. 2 of them came to be deceased after the vaccination. And in this figure B, we can see the results of the exome, and it's an illustration that showed that the patient presented homozygous to case 2 and 3 and 4 and 5. And we can see also the axons where we have the late proband comparing to the other family members. And here we have a modeling, molecular modeling, and we can see the complex of interferon with its receptors in healthy individuals. And here in our probands, these results were obtained with the collaboration of Dr. Zilton Vasconcelos. And continuing on the functional analysis, in this first graph, we can see graph A, we can see the profile of the culture, and we can see in purple the results of the adverse events. So we can see an increase in the production of beta interferon and also at L1 beta, which is an anti-inflammatory protein. This happened when we analyzed the cell profile, and we can see that the patients in purple, when compared to the control, they present a higher rate of the complex. And we also have the presentation of also presented the cytotoxic profile. You can also see the patients with naive cells for CD4 and CD8 that are much higher than the controls, the healthy controls. And we evaluate the memory. They do not have specific memory for yellow fever vaccination. And going to our transcriptomic profile that were obtained in partnership with Dr. Otávio Marques from the Federal University of São Paulo. And you can see the healthy population for control and the adverse event population. And they had different genes that were expressed differently than the cases of adverse events. And we got these that were regulated in the cases of adverse events. And these genes are related to the pathways that are pre-inflammatories, and you have these several enzymes and the signalization of type 1 interferon. And you can see individually the genes that are more regulated, related to the inflammatory response and the interferon receptor itself. And as conclusions, we have that the adverse events associated post-vaccination of yellow fever, they are related to type 1 interferons and the reaction with its receptors. And it's not clear to us if this happens a down regulations or up regulation in the pathway of regulation of type 1 interferon. But we know that these cases present an innate response that is dysregulated, and also some kind of problem in the development of memory for yellow fever. An additional information is that we have these patients that followed that with a specialized medical care. And this is one of the advantages of this project. As a perspective, we have these studies of the functional aspects of these genetic errors. They can help the developments of therapeutic strategies and alternative for vaccines such as mRNA, where you do not have viral replication, and it can also help in the reduction of vaccine existences by fighting against misinformation, vaccine fake news. Thank you very much.